Well, hello there. Today we are going to start talking about the macromolecules. These are the large molecules that combine to form cells. Get my pen going here. So we're still talking about molecules like we have been. We're just talking about big ones. So macro means large. And your book just calls these biological molecules, which is also accurate. Okay. So there are four classes or types of macromolecules, and we're going to talk about all of these. There's carbohydrates, proteins, nucleic acids, and lipids. In this lecture today, we're going to talk about carbohydrates and proteins, and then I'm going to make a second lecture about nucleic acids and lipids. Okay, but today first we'll start off by talking about just some general things about many of the macromolecules. Okay, and the first is that all of the macromolecules, so all four classes of macromolecules, are composed of a carbon backbone or a carbon skeleton. And here are some examples of carbon skeletons shown here. So carbon skeleton are just a bunch of carbons that are added together, and they can form many different shapes. Carbon is very important because it is so versatile. So remember, carbon, the electron shell diagram is shown here. Carbon has 6 electrons. Okay, first 2 are in the first shell, and the last 4 are in the second shell, or the valence shell. Okay, and since that shell can hold 8, 4 are missing. So you could think of them as having little spots at next to each one of these electrons for another electron to bond, or an opportunity for a covalent bond would be another way to think of that. And carbon is more versatile than any of the other 3 elements that we're talking about, right? Because it can make those 4 bonds. It could make 4 single bonds, or it could do combinations of things. It could have 1 double bond and 2 single bonds, 2 double bonds, even 1 triple bond and 1 single bond. Any combination is possible as long as carbon has 4 bonds total. You can even have a carbon-carbon quadruple bond where there's 4 bonds between 2 carbon molecules, but you'd have to make that in the lab, not in real life. Okay, so these are all different shapes that can be formed by carbon backbones or carbon skeletons, and we will see these as we go through. Of course, we also see a lot of hydrogens here. And we do see a lot of hydrogens in the macromolecules, but that's not what the carbon skeleton is talking about. Okay, in addition to having the carbon skeleton, we also find a lot of functional groups found in these biological or macromolecules. Okay? So the first one we'll talk about is the hydroxyl group. I often refer to this as just an OH group. Okay? So those two things mean the same thing, and this is simply an oxygen bonded to a hydrogen, right? And then that oxygen has to be bonded to something else, and it's frequently bonded to a carbon, like is shown here. So here's a hydroxyl group, hydroxyl group, hydroxyl group, hydroxyl group. So we saw these yesterday, or I guess that would be Tuesday in lab, in the activity, excuse me, when you guys were building some of those molecules. Okay. Oh, and I should say, I forget to say, these are examples of carbohydrates. We see lots of OH groups in carbohydrates. These are examples of amino acids, and amino acids are what we use to make proteins. In amino acids, we could definitely potentially find some OH groups just hanging out there by themselves. But what I want to call to your attention here are 2 others, the carboxyl, and I often just call this the carboxylic acid group, and the amino group. Okay, so every amino acid has an amino group on one end, so the NH2, and this one has one here too, and then a carboxyl or a carboxylic acid group here. Okay. I know this is showing it as an O-. This is simply become ionized, and is the ionized form of the OH, carboxylic acid. And so those are we often see those in proteins. And then the last thing I want to call your attention to is the phosphate, and we usually call this a phosphate group. Okay? The phosphate group is a phosphorus. Phosphorus is a different element that we haven't talked about yet, and it's not super common in life forms, but it is common enough to be important. especially because it's in this molecule here, which is ATP, which is cellular energy. And we'll come back and talk about this later. So a phosphate group is a phosphorus bonded to 4 oxygens, and we see them here. So each one here is the phosphate groups, and we see them also in nucleotides, which we'll be covering in the second macromolecule lecture. All right, carbohydrates, proteins, and nucleic acids are all polymers. Lipids are not polymers, so they don't have this in common that we're going to talk about right now, but carbohydrates, proteins, and nucleic acid are all polymers. And polymers are simply large molecules that you make by linking together many subunits or monomers. Okay? So a polymer is a large molecule composed of many monomers, and the monomer you could also think of as a subunit. And a really great analogy for this is thinking about them as bricks or as Legos. Okay, we have a lot of Legos in my house since I have kids, so this one makes a lot of sense to me. Monomers are the subunits, and you could think of these as the single Lego bricks, like these little ones that just have 2 little dots on them. You can link them together, of course, and when you do, you're forming a polymer. So as long as you put 3 together, you now have what's called a polymer. Okay, you can add more, of course, and make a bigger polymer, and even more and make an even bigger polymer. Okay? And this is the idea for the carbohydrates, the proteins, and the nucleic acids. We take a monomer, and we're going to talk about what the monomers are, and we link it with other of those monomers, and we stick them all together to form these really big molecules that are polymers that just have repeating subunits over and over again. Okay, so now we're going to talk about how do we put those subunits together. So that'd be like taking 2 Lego bricks and putting them together to make, this would actually not be polymer yet because it would have to have 3. This would be like a die Lego, 2 Legos. But we're just going to talk generally about how do you put monomers together. Okay, all monomers are put together through this process called dehydration synthesis. This is a type of chemical reaction. Okay, so just quickly about reactions, this here is showing a Lego synthesis reaction, and this is showing a dehydration synthesis reaction. Both of them show reactions. On this side over here we have the reactants, and over here we have the products, and this arrow indicates that a chemical reaction is happening. So we will show this over and over again this quarter, and so you're just going to have to get used to that kind of graphic depiction of chemical reactions occurring. Okay, so what do we have here? We have, this is, let me see what my next slide is. Okay, oops, I took off some things. Okay, so this is a monomer, and this is a monomer. Okay. These are shown very generally to just describe what dehydration synthesis is. Okay, so dehydration synthesis is a type of reaction that links 2 monomers together and removes water. Okay, so let me get this pen going and change my color. Blue. Okay, so we want to link these 2 together, and when we do, we're going to take the hydrogen off of one and the OH off of another one, because they all have these OH groups hanging off of them at just the right places. And that's going to be taken off and made to form water. Okay, this is why it's called dehydration, because you're pulling water out. Okay, when this hydrogen and this OH group come off, then a new covalent bond is going to form between this oxygen of this first monomer and the actual monomer of the other one, right? So we're going to be making a new bond here, okay? And that's what is shown here. And now those 2 monomers are linked together. This just has 2, and of course when we are talking about building polymers, we want to put many together. But that process just keeps happening over and over again, where you take one monomer, do this dehydration synthesis, pull water out, and then have that monomer linked to it. And then again, this is the new bond. Okay, depending on what type of polymer we are talking about, the specific name for that new bond that is created by dehydration synthesis will be different. And for some of them we'll talk about what that name is. Okay, and then in the end, you would have, this would be a polymer, right? Okay, and this was made by taking a bunch of monomers and linking them together through dehydration synthesis. So when, let me just number these, so this would be 1, 2, 3, 4, 5, okay? When monomer number 1 and number 2 were linked together, then water came out. So every time you link two monomers together, you water comes out. Okay? And then you would have this one with 2, and you want to add another one, and again, water would come out. Okay? And when you link, then you would have 3 together, and you want to add another one, and again, water would come out. And then finally, you would have 4 stuck together and want to add another one, and then dehydration synthesis would pull water out again as well. So this process that's shown here, just between 2 single monomers making a dimonomer, I guess, for lack of a better general word, would happen over and over again, okay? And we keep getting formation of new bonds that are then going to link those monomers together, okay? So that's the general idea with dehydration synthesis, and we're going to be practicing this in our activity in class on Thursday. Okay, a couple other things about polymers is that they can be composed of many of the exact same monomer or multiple different monomers, and I just want you to just kind of put this information in the back of your head and store it, and then as we go through the different polymers, the different macromolecules that are polymers, we'll talk about that. And the same with this. You can have different patterns of attachment between the monomers and create different specific polymers of different molecules. And we especially see this when we talk about carbohydrates or carbs. Okay. And then each polymer, and right now we're talking about carbohydrates, proteins, and nucleic acids, has its own specific monomer. We are going to go through today and this lecture and in the next lecture and talk about what the specific monomers are and talk about how they're linked together to form the polymer. Okay, so but this, you need to just memorize this to get these names put together. Okay, so this is the polymer and this is the monomer. You can think of these as polymer monomer pairs. So carbohydrates, carbohydrates go with monosaccharides. So you take a bunch of monosaccharides, link them together, and you come up with a carbohydrate. If you take a bunch of amino acids together and put them together, then you come up with a protein. And if you take a bunch of nucleotides and put them together, you get a nucleic acid. So you definitely want to memorize these in addition to like knowing what they look like. All right. So as I said, today we're going to go through carbohydrates, and we are also going to go through proteins. Okay, so it means we're going to talk about monosaccharides, and we will also talk about amino acids. Carbohydrates you might also know as the names of sugars or carbs. A lot of the foods that we like to eat are full of carbohydrates, and often we're trying to limit the number of carbohydrates that our bodies get. You know, these ones here would be kind of, you know, junk food, simple carbohydrates full of simple sugars, things that are called monosaccharides and disaccharides. Whereas this here, this is whole wheat bread. It contains more complex carbohydrates, what you would also call polysaccharides. Okay. Our cells, so I know many of us are trying to limit the number of carbohydrates that we have, but you don't want to get rid of them entirely because they're really important for our cells to function. Okay. So one of the things that our cells does is take carbohydrates, specifically glucose, and feed it into this process of cellular respiration. So I'm just going to write CR, cellular respiration, to make ATP. Okay, we are going to learn all about this later in the quarter, but ATP is cellular energy. Our cells use this to do work inside of the cells. And cellular respiration is this long, complex process that takes the energy in the sugar and puts it into ATP. Okay, so if your body doesn't have any carbohydrates, then you wouldn't have any ATP. That would be bad. Carbohydrates can also be used for energy storage. and also for structure. Okay. And then carbohydrates are the only macromolecule or the only polymer that we talk about the three size classes. And so that can make it a little bit confusing. So there are monosaccharides. These are single sugar subunits. So mono means one, and saccharide means sugar. Okay. This is the simplest sugar, and we'll go through examples of this. There are multiple kinds of monosaccharides. But they do exist, and they do things, and particularly they're used for cellular respiration inside the cell. And then there are disaccharides. So di means two, and saccharide again means sugar. So this would be two sugars or two sugar subunits. And then finally there are polysaccharides. Poly means many sugars. So monosaccharides and disaccharides are often what we think of as simple sugars. Okay. This actually has starch in it, so it would be complex sugar. Definitely cookies have plenty of disaccharides in it and make it a simple sugar. Polysaccharides are more complex carbohydrates, things like starch and cellulose that have many sugar subunits stuck together. Okay, there are 5 common monosaccharides. Okay, a monosaccharide is a single sugar subunit, and this is, remember, the monomer. So monosaccharide is the monomer that goes with the carbohydrates or the carbs. Okay, that just goes back to that chart I showed you. Okay. Another way to think about this would be it's the single brick that's going to be used to build bigger ones. Okay, so there are 5 common ones. Let's see. First of all, I just want to show you something about how these are written. Okay, so this is an abbreviated form. It shows a ring structure, and this next slide shows you the abbreviated structure. Okay, so this is glucose. Glucose you can draw in a couple ways. Okay, this is has the most information in it. Right, it shows you where each of the atoms are, and it shows where most of the bonds are, with the exception of this OH group. So when you see an OH group, it really means O bonded to a hydrogen. So they've condensed that, okay? But it definitely shows all the carbons, right? And you see this carbon ring, okay? This would be that carbon skeleton, right? And sometimes we see an oxygen in it, as we do here, okay? But that's the carbon skeleton. And so since that's the carbon skeleton, and we see it often, it is frequently abbreviated like this, okay? And when you see it drawn like this, each corner represents a carbon atom, right? So that was shown here. So you can think of a carbon being here, 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 and here, okay? But taking the carbons out just makes it a little more easily understood. Okay, this one over here is even further abbreviated. We have each corner representing a carbon, and then we're also missing the hydrogens. So they took those off because they just kind of get in the way. When you're looking at structures, and you know that a carbon is here because there's a corner there, you need to remember that, okay, carbon can make 4 bonds, right? So if I see that carbon there, and this is showing me that it's bonded to another carbon here, and to another carbon here, and then that carbon right here, these are the same carbons, is bonded to an oxygen. Okay. That oxygen is bonded to a hydrogen. Okay. So carbon has to make 4 bonds, right? So it has this little electron there, oops, right here, and if it's not bonded to anybody, it'd be reactive. So they've taken off the hydrogen, and then we just have to remember to assume that there's a hydrogen there as well. Okay, so there's a hydrogen here, and here, and so on around the ring at every corner. There is a hydrogen that they took off, right, just for simplicity's sake. Okay. So let's go back to look at our monosaccharides again. So these are shown in that abbreviated form in the middle, right? It's showing the hydrogens, but it's just showing the carbon skeleton as lines, with each corner representing a carbon atom. Okay, so there are 6, like I said, 5 common monosaccharides. 3 of those are called hexoses. Hex refers to 6, so these are the 6 carbon sugars, and if you count each of these, you'll find that they have 6 carbons, right? So 1, 2, 3, 4, 5, 6 carbons. 1, 2, 3, 4. Oh, I forgot one. 1, 2, 3, 4, 5, 6 carbons in fructose, and 1, 2, 3, 4, 5. Oh, and this guy's sitting on top of that. I didn't notice I put on. There's also a CH2OH group up there. My little Lego guy is in the way, so just pretend that's not there. So there's 6 carbon on these also, and you'll notice that these all have like a striking similarity to each other, right? So there's not a huge number of differences between them, but they are considered to be different. They are different molecules. They have different shapes, and so they are different molecules. Okay, and then there are the 5 carbon monosaccharides, or the pentoses. Pent refers to 5. Okay, and that's deoxyribose and ribose, and these each have 5 carbons. 1, 2, 3, 4, 5, and then 1, 2, 3, 4, 5 there as well. Okay. So let's take a closer look at just the 6 carbon sugars. Definitely the most common one that we'll see is glucose, but you guys are also going to be building galactose in your activity on Thursday. Okay, and there, now I've moved that Lego thing, so now you can see that CH2OH. Okay, so as you count these, I just wanted to count the carbons and count the number of oxygens and hydrogens. You will find that these all have the same exact chemical formula. Okay, so the chemical formula for all of these is C6. Okay, that means they all have 6 carbons, as we counted before. For hydrogens, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 hydrogens, and that would be the same for all of these. So let's just do this quickly. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And on this one, it doesn't show those hydrogens hanging down. You'll notice that. So there's 12 hydrogens, and then oxygens, there should be 6 oxygens, right? So 1, 2, 3, 4, 5, 6. Okay. They all have the same general chemical formula. Okay. So if you just wrote C6H12O6, and unless you told someone you were talking specifically about glucose or fructose or something, then they wouldn't necessarily be able to tell. Okay. That said, the most common monosaccharide that we'll talk about this quarter is glucose. And like when we're talking about photosynthesis and whatnot, we'll just refer to it as C6H12O6 without noting that it's glucose, because we're just assuming that. Okay. I also just wanted to remind you the uses of monosaccharides, of the 6 carbon monosaccharides. Let's see. No, okay. I did not write it down, but I did note it before. But the uses, just write it again, are immediate energy use. Okay, and that's in cellular respiration that is used to make ATP. And then these are also used to build other molecules, to build disaccharides and to build polysaccharides. Okay. Another important thing to note, so here's glucose again, okay, is that we number the carbons. So remember, in this structure, each corner is a carbon. Okay. And then it's really handy to be able to talk about which carbon you're talking about, so that 2 people could refer to the structure and talk about, say, what's attached to one of the carbons. Okay, and that's why we number them. So this first carbon here, and we always start to the right of this oxygen that's in the ring. Okay, the first one is number 1, the second is number 2, 3, 4, 5, and then number 6 is always the one that's sticking out there by itself. Okay? And we'll practice numbering these in our activity, but it is really important. That way I could say that in glucose, on carbon number 1, the OH group is pointing down, and on carbon number 2, the OH group is pointing down. Carbon number 3 is pointing up, etc. Okay, and then here are the pentoses, the 2 5-carbon sugars. Okay, and these are deoxyribose and ribose. Okay? They have 5 carbons. They actually don't have the same chemical structure because they have only one major difference, and that's in deoxyribose, there's just a hydrogen here, and this is carbon number 2, and in ribose, there's an oxygen and OH, and that's the only difference. So this is ribose, this is deoxyribose, meaning it's missing one. Deoxyribose is used in building DNA. So we find this in the DNA nucleotides. Okay, so we will come back and talk about that guy later. And same with this, we'll see this one again because we find it in the RNA nucleotides. Okay, and of course nucleotides, if you remember from what I said previously or from your reading, are used to make nucleic acids, which is one of the type of macromolecules that is a polymer. Okay, so those are the 5 types of monosaccharides. Really, we're not going to see these guys, the deoxyribose and ribose, again until we talk about the nucleotides, and we'll just focus on these guys, on the monosaccharides that are 6 carbon sugars. Okay. Oh, I'm sorry. I did want to tell you one other thing though, and we'll come back and talk about this later again too. But we also number the carbons in the 5 carbon sugars in the pentoses. We do it only slightly differently. We still start with the oxygen and go to the right, and that's carbon number 1. But because it's in a pentose sugar, we say it's 1 prime, and we put a little prime there. And then 2 prime, 3 prime, 4 prime, 5 prime. And we do that on both of those. Okay, so note that because it will become important later this quarter. All right, so we talked about monosaccharides, and now we're going to talk about disaccharides. Disaccharides are what you get when you take 2 sugar subunits and put them together via dehydration synthesis, and I'll show you in a second how that happens. These are used frequently for immediate energy use because it doesn't take a cell too much to break this bond here and release them into their monosaccharides, and it can go right in into cellular respiration. And it's also a way to transport sugar around. So for example, trees, when they're doing photosynthesis, they make glucose and then they quickly put that glucose, they move things around a little bit and make a disaccharide to move that around the cell. What I do want to talk about is how these monosaccharides are linked together in using dehydration synthesis. Okay, so we're going to be looking at just these 3 monosaccharides, glucose, fructose, and galactose, and they're shown by different symbols here. And those are used to build disaccharides. Okay, remember di means 2, so 2 sugar subunits. Okay, and they're linked together via dehydration synthesis. Okay, so this is similar to what I showed you before. What I showed you before it was very general, right? So we just said 2 monomers were linked together by dehydration synthesis. Right now we're talking about 2 specific monosaccharides being put together by dehydration synthesis, but the same is still true. So an OH comes off of one and hydrogen off another, and those 2 things together form water. So water is pulled out, and then a new bond has been formed between those, and now you have them linked together. Okay, so that's how you'd form a disaccharide, is to put 2 of them together through dehydration synthesis. So every time you take 2 monosaccharides, put them together, then you get 1 water molecule. And here is shown sucrose. Sucrose is table sugar. Sucrose is a disaccharide formed by connecting a glucose and a fructose through dehydration synthesis. There's another one, maltose, is formed by putting 2 glucose monomers together, or 2 glucose monosaccharides together, pulling water out and linking them together. Okay. Of course, there are another type of carbohydrate, which are the polysaccharides. Those are the ones that are really long. Okay, and this is actually would be a short one, having only 6 subunits, but they're put together the same way. Okay, so monosaccharide is just 1, disaccharide is 2 linked together using dehydration synthesis, and a polysaccharide is 3 or more monosaccharides, and they can be any type of saccharide linked together using dehydration synthesis. Okay, so polysaccharides, 3+ sugar subunits, they are used for either energy storage or structure. Okay, and they are made using dehydration synthesis. Okay, so essentially you would take a disaccharide and you're going to add a monosaccharide to that. You're going to pull out water, and now you're going to have this new linkage where you have this new bond formed. Okay, if you wanted to make a longer polysaccharide, then you would just keep doing that, adding another subunit, and pulling water out. And indeed, the polysaccharides are very large, okay? So there's 3 polysaccharides that are common. The first is starch. Starch is composed of a bunch of glucose monosaccharides linked together with a somewhat branched kind of structure. So if you remember, I said that you could link together the same subunit but with different patterns and come up with molecules, and that's what we see when we look at the 3 polysaccharides that we're going to examine: starch, glycogen, and cellulose. They're actually all built using glucose, okay, but the glucose monosaccharides are arranged slightly differently, creating different molecules. Okay. Starch is used by plants for energy storage. Okay. So we looked at a banana cell, and remember we found the amyloplasts. The amyloplasts store starch in them, and that's why they turned purple, because we added the I2KI to make it purple so we could see them. The banana has lots of starch in it because it's a good food source, right? So the plant is storing that starch as energy. From the plant's point of view, it's not trying to make food for us. It's trying to make, well, that's not true. It's trying to make food for us to get us to eat the plant and release the seeds. So, of course, that doesn't happen with bananas anymore. Bananas don't even have seeds, modern bananas, but the ancestors of the bananas that we eat had seeds, and the reason the fruit evolved to carry starch was to get an animal to eat that fruit, and then the seeds would pass through the gut of the animal and be deposited elsewhere and planted, and that would help propagate the species. So from a plant's point of view, it's either going to add starch to something to get an animal to eat it, or put it into some storage device, say like potatoes, so that that plant can then use that starch later to grow. Okay. From our point of view, from a heterotrophic point of view, starch is a great food source. We can digest it, and we're going to talk about that later, but we chop off glucose monomers one by one and send those into cellular respiration and get lots of energy out of it, get lots of ATP out of it. And then there's glycogen. Glycogen is also a single sugar subunit called glucose. So glucose is the monosaccharide. And this is highly branched. So when we looked at starch, it's moderately branched. Okay, so they're all glucose, but it's moderately branched. These are all glucose, but they're more highly branched. Glycogen is found in animal cells, primarily in the liver and in muscle cells. And animals use it for kind of mid-term energy storage. We use fat for long-term energy storage, but we keep a store of glycogen in our liver and in our cells for our body to use if we run out of, food that we have recently eaten. And so that's what that is. Okay? And then finally there's cellulose. Cellulose again is composed of glucose monomers linked together, and in this it's in these long unbranched chains. Okay, and this forms cellulose, which forms cell walls. Okay, so here's an Elodia cell that we looked at on Tuesday in lab. Okay, and remember, every plant cell has a cell wall, and that cell wall is made out of cellulose. I said before that the cell wall was just a sturdy structure, and that's what it is. It's made out of cellulose. These long strands of cellulose together form a very sturdy structure. Okay. So those are all polysaccharides. I want to show you this little video about dehydration synthesis, basically. And you can adjust the volume on yours. I don't want to make it too high.
Polysaccharides are all polymers, which are very large molecules made up of simple sugars. They include starches, glycogen, and cellulose. We'll see how these are produced or synthesized. Polysaccharides are made up of many smaller molecules called monomers joined together. One common monomer is glucose. Glucose is a simple sugar or monosaccharide. Two other common monosaccharides are galactose and fructose. You can see these all have slightly different shapes or structures. A molecule of glucose contains 6 carbon atoms, which are numbered like this. These 2 OH groups are used to bond glucose monomers to each other. We'll see how that works. Here are 2 glucose units. we'll consider this OH on the first glucose molecule and this H on the second glucose molecule. An enzyme in plants enables two glucose molecules to join together. Let's have a look at this. We can imagine the H and the OH coming off and the two molecules moving together. We should point out here that the actual mechanism for this is much more complex and involves enzymes. The simplified animation here just helps us visualize how the two glucose molecules can bond together. When two glucose molecules bond together, they form another sugar called maltose. Because maltose is a combination of two glucose monomers, it is called a double sugar or disaccharide. The H atom from one glucose bonded to the OH from the other glucose forms a molecule of water. Because a larger molecule is made from two smaller molecules, this process is called synthesis. And because a water molecule is released, it is called dehydration synthesis. Another disaccharide is formed when a molecule of galactose and a molecule of glucose bond to each other in a dehydration synthesis to form lactose. Lactose is a common disaccharide found in milk products. Still another disaccharide is formed when a molecule of glucose and a molecule of fructose bond to each other with a dehydration synthesis to form sucrose, a common disaccharide found in table. Now we'll go back to our molecule of maltose and see how we can add more glucose to it to form a polysaccharide. More glucose molecules bond to this maltose and grow this chain by the process of dehydration synthesis, eventually forming starch, which consists of very long chains of glucose units that have some branching. Starch molecules can consist of up to 4,000 glucose units. Glycogen is another polysaccharide. You can see it has a lot more branches than starch. It is produced in animals rather than plants. When blood levels of glucose are high, the liver produces glycogen, which is a way of storing glucose for when it is needed. Glycogen is stored in the liver and in the muscle cells. Like starch, glycogen also consists of long chains of glucose units. But in glycogen, the chains are much more highly branched than in starch. A different type of polysaccharide called cellulose is produced in plants. Cellulose consists of long, unbranched chains of glucose units. Notice that the linkage between glucose units is different in cellulose than it is in starch or glycogen. In cellulose, the oxygen atoms between the glucose units alternate their positions, whereas in starch and glycogen, the oxygen atoms between glucose units are all in the same relative positions. It is important to note that the human body is capable of digesting starch and glycogen, but it is not capable of digesting cellulose. The body breaks down starches and glycogen in steps, eventually ending up with many single glucose molecules, which the body uses for energy. Even though the polysaccharide cellulose is not broken down by the body, it provides the body with fiber, which is believed to help with digestion in the large intestine.
Alright, this is just boring. Let me get myself situated again. Alright, and get rid of that. Go. Now go. Go. Okay. Righto. Okay, so that was macromolecules. Carbohydrates. Excuse me. That was carbohydrates. And hopefully that video, I know he has a kind of low voice and kind of, there we go, boring sounding. I think he does a good job explaining it and putting it together with the animation, so hopefully it kind of capped what we just talked about. Okay. Next, we're going to talk about proteins, and like I said, that will be the last molecule that we talk about today, or in this lecture, and then there'll be another one for the nucleic acids and the lipids. Okay. So proteins are really important inside cells because they do all of the work or jobs inside cells. And this little cartoon shows just some of the things that proteins do for us. Okay? So proteins provide structure. For example, let's see, to my pen, structure such as hair and nails. Okay? They're also in muscles, which you could consider part of your structure as well. Like, we wouldn't be able to stand up if we didn't have muscles that are attached to our bones. Also, the cytoskeleton that is not listed here provides structure for our cells and allows things to move inside our cells. Movement, so muscles is our big one. Muscle proteins allow muscles to contract and allow us to move. Hemoglobin allows oxygen to be carried around inside our blood, delivered to different parts of our body. Signaling is a major thing that proteins do. Okay, so our cells have on them inside the membrane, as membrane proteins, we have proteins that are receptors that can capture signals that are also proteins. Some of them are hormones that are going through our body, and those receptor proteins get the signal, and then a cascade of events is set off from that. So that's really important in a way that our different parts of our body can talk to each other. And then also brain and nerve cells use signaling. So there's some ion channel proteins that allow the movement of ions across the membrane that allow for that signaling to happen. So that's really important. When we talked about that white blood cell that was chasing the protein, or chasing the bacteria, we were talking about antibodies. Antibodies are a type of protein that are for defense, okay, and they're made by white blood cells. Proteins sent out and attack the invader and mark it for destruction. And then finally, catalyzing reactions. So we'll be talking all about enzymes later, and then this is kind of talking about enzymatic reactions too. Catalyzing reactions, of which there are millions, to make them happen in order for cells to do everything that they do, we have to have biochemical reactions happening, and proteins are responsible for that. So without proteins, our cells wouldn't be able to do anything. So proteins are really important, and you know, it's pretty geeky to have a favorite macromolecule, but of course I do, and those are proteins. And one reason is just because they do so many cool things, and then also their structure is really cool too. Okay, so proteins are polymers, and they are polymers of amino acids. Okay, so this is the monomer that goes along with the proteins, and the protein, remember, is the polymer. And that again goes back to that chart. Okay, right over it. Good job. So there are 20 different amino acids. That's what that says, 20 different amino acids. And although there are 20 different, they all have the same 4 basic parts, okay? And so we're going to go through that. So this is one amino acid, showing a basic amino acid structure. They all start out with an amino group, okay? That's one of the functional groups that I pointed out at the beginning of the lecture. And then they have a central carbon. It's called the central carbon because it's in the middle. Everything else is attached to it, okay? And then there's the carboxyl group. This is also the carboxylic acid group is another name for it. It's that carbon double bonded oxygen, OH group, which is another one of the functional groups that I pointed out at the beginning. And then finally, the fourth thing is the R group. So R stands for variable in chemistry. So that just means that this is the part of an amino acid that's different. And remember, there are 20 different amino acids, so they all have a different R group. All 20 have the same amino group, the central carbon, and carboxyl group, but have a different R group. And I'll show you those in a moment. I just want to point out also that the central carbon always has this little hydrogen hanging out here. The hydrogen doesn't have a name, but it always goes with the central carbon. And before I show you the chart showing all the different amino acids in the R groups, I just want to point out this, and that's that amino acids will ionize in water, and when they do so, the amino group will pick up an extra proton and become positively charged, and H plus equals a proton, okay, and that's because a hydrogen, right, a hydrogen atom just has one proton and one electron, If it's ionized, it loses its electron, and then it has just one proton, right? That's a good thing to know, that a hydrogen ion, H plus, is the same thing as a proton. Okay, so it picks up a proton and becomes ionized, and then so the amino group takes on a positive charge. We still call it the amino group. And then the carboxylic acid does the opposite. So it acts as an acid. Acids donate hydrogen ions or protons to their solution, and that's what happens. So the proton comes off and leaves the electron with the oxygen, and it becomes negatively charged. And we still call this the carboxyl group or the carboxylic acid group. It's just charged. Okay, so this is ionized and this is non-ionized. And since water is everywhere inside cells, we typically find the amino acids ionized in cells. And some of the diagrams show that and some don't, so I just want to make sure you're aware of it. Okay, so this chart here shows the 20 different amino acids. Okay, I definitely don't expect you guys to memorize these, what the different R groups are, but I do expect you to know and be able to figure out the basic structure, right? The amino group, the central carbon, the carboxylic group, and then here, the R group is shown in white. Okay, so wherever the white box is, that's where the R group is. Okay, and remember the R group is the variable region, so that's the part that's different. And if you compare these, all 20 of them will have the same exact amino group, right? No matter which one you look at, it's H3N, and that's just the same as writing it out like this with a little plus. Okay. That's the abbreviated form of that. Okay. Oops. And then they all have the same central carbon with a hydrogen attached, and the R group's attached to that, and then they have the carboxylic acid group that's the same for all of those. Okay. So that's all the same. The only thing that's different is the R group. Okay. And the R group, as you can see, has a lot of different possible shapes and a lot of different possible configurations of different atoms. Okay? These are put into different boxes based on the chemical characteristic of the R groups. Okay? Each R group can be either hydrophobic, and that's what these ones are here. They're hydrophobic. If you look at them, they have mostly carbon-carbon and carbon-hydrogen bonds that are nonpolar, and that makes these act like oil. So these are the hydrophobic R groups. We also have some that are hydrophilic. Okay, they don't have charges on them, but they have lots of polar bonds, so they have to have lots of partial charges on them. So they're polar, and they're hydrophilic. They can make hydrogen bonds with water. And then there are two groups that are actually ionized. These R groups have a positive charge, and these R groups have a negative charge. Okay? And those ones, of course, can interact with water as well. Okay? So all of these ones you would put into the broad category of hydrophilic. Okay? So if you put them in water, they'll like to make lots of hydrogen bonds. And all of these ones over here in this group, you could put in the broad category of hydrophobic. Okay, their R group will act like water. Okay, and we're just talking about the R group because, of course, the amino acid itself is charged, right? So it always can interact with water when they're free like this. Okay, the other thing I just want you to notice about this is that each amino acid has its own R group and then has its own name. Okay, so the name is always associated with the R group, and again, I don't expect you guys to memorize that or to memorize which ones are hydrophobic and which ones are hydrophilic or anything. I just want you to recognize that there are 20 different amino acids. They each have their own R group and they each have their own name. They have a long name and then they have a 3-letter abbreviated name. Okay, and we will be using these throughout the quarter. So you need to be able to find your way around this chart. All right, so to build a protein, dehydration synthesis is used again to connect the monomers, in this case amino acids, together. Okay, so again, OH from one and a hydrogen from another is going to be taken off and lost as water. Okay, and that's shown here as well. And then a new bond will be formed, right? So right here is where a new bond will be formed. You're going to take one hydrogen off that nitrogen, so it will want to make a bond, and an oxygen off this carbon, so it will want to make a bond, and so it makes a new bond. And of course, this doesn't just like magically happen. It is carried out by enzymes, and we'll talk about that later. And then you get what's called a dipeptide. A dipeptide has two amino acids linked together through dehydration synthesis. This new bond that is made is called a peptide bond. A peptide just refers to a protein. It's a word that refers to protein. And so another way to think about that would be a protein bond. Just because it has a name, it doesn't mean it's a different kind of covalent bond. It is simply a covalent bond between a carbon and a nitrogen, but it connects the two amino acids, right? So you could take on here and circle one amino acid, right? That's one amino acid. It has its amino group, its central carbon, and its carboxyl group. Because it is part of a longer molecule, a dipeptide, it has lost its OH group. But this is still the carboxyl group right there. That's still the carboxyl group. And it has an R group. And in here, just the R group is just shown as R, not as showing its whole structure, just for simplicity's sake. Okay, so there's one amino acid, and here's another amino acid. So a dipeptide are two amino acids linked together. Dipeptides are not the same as proteins. Proteins have many amino acids linked together. Okay, so here's something that I drew primarily because I couldn't find a good diagram for it. So hopefully you guys can read my handwriting. Okay, so this is starting here with a dipeptide, right? Just like what we started with, what we ended up with on the last page. Okay, here's one amino acid, and here's the second amino acid in our dipeptide. If we are going to take that and build a protein out of it, we're going to add another amino acid, and that's what's shown here. Another amino acid is coming in, and dehydration synthesis is going to remove the OH and the hydrogen, and water will come off, and then you will have a tripeptide, and now you have a very short protein, right? You have a very short, or you might call it a polypeptide. And it has three amino acids, right? So here's one amino acid. And again, it has an amino group, central carbon with an R group, and a carboxyl group. Here's a second amino acid, and a third amino acid. Okay. And then this process would just keep going, right? So if you want to make a longer protein, you would add another amino acid through dehydration synthesis. So again, water will come off, and then you would end up with 4 amino acids linked together. And then again, you could circle them. So here's one amino acid, 2 amino acids, 3, and 4. Okay. Proteins are actually hundreds or even thousands, tens of thousands of amino acids long. So this process just keeps going and going until the whole protein is built. Okay. And then here's a drawing, right, shown of a short protein just with 3 amino acids, just circling the different amino acids. That's definitely something that you guys need to be able to do. And then in the end, you end up with this long chain, this long protein that is made of hundreds or thousands of amino acids linked together. Okay, this is a good way to start looking at it because we're going to circle back to talking about protein structure when we start talking about enzymes, and we'll talk about protein structure more in depth in I don't know, a week or so. So what this is showing here is each circle is representing an amino acid, and it has its little 3-letter name back from that chart when we were looking at the different amino acids. It doesn't show the R group because that would just get so busy in here, but it has the name on it, okay? And each one of these circles represents an amino acid, right? So it has an amino group, it has a central carbon, and this is showing the R group pointing down, but the central carbon still has a hydrogen, and then has a carbon with the double bonded oxygen, so that's the carboxyl group. Okay, so even when they are attached to each other, you can still point out those 4 basic parts. Okay. So there's one amino acid, that would be glycine, and then there's proline would be here, still has an amino group. This amino group here, it's missing one of its hydrogens, but it's still called an amino group. It's just lost one of its hydrogen in that hydrogens in the dehydration synthesis. Okay, and so it's shown here, and then let's 3 of them, right? So, but you could do that for all of them. You could pull it out and look at the structure, but it would start to get very busy. Okay? And so it just, shows the order of them, whatever it would be, would make that protein. On the protein, there's an amino end. That's the end of the protein that has the amino group sticking out, right? And that's what that is. And then on the other end, you could call it the carboxyl end or the carboxylic acid, and that has the carboxyl group sticking out on that end.